Hey friends, it's been a while since I put up a video. My excuse is that work has been a bit frantic lately, and I was off giving a talk for Darwin Day a couple of weeks ago at Humanists of the Palouse, and I'll include a link to them. They're a good group. You should support them. And that threw off my whole schedule. But hey, I've got a little time today, so I thought I'd give you a greatly trimmed down version of the talk I gave in Moscow, Idaho a couple of weeks ago. My subject was epigenetics. This is always an awkward subject for me because when I put on my developmental biologist hat, I have to enthuse over it and say how essential and important it is and how much really cool work is being done in the field. This is an important topic. But when I put on my evolutionary biologist hat, I have to instead get kind of cranky and tell you that the claims are vastly overinflated, and no, epigenetics is only relevant to evolution as a splendid example of things like plasticity and as part of the mechanism behind norms of reaction. But as a purely evolutionary process, it's transient and relatively unimportant. So it's intensely uncomfortable to sit here just oscillating between these two ends of the spectrum. But I kind of have to, because there's a lot of crap science being promoted as interpretations of epigenetic phenomena right now. For example, I have to mention James Shapiro's third way of evolution, which gets so wrong. This is from Shapiro's website, and I'll include a link to it in the description in case you don't believe me. People are using epigenetics to argue for a Lamarckian version of evolution. This is utter nonsense. You may also notice from his example that he's arguing for teleology, that somehow pandas knew how to modify a wrist bone to make a new thumb, and that they somehow worked out how to build one. There's a real effort to impose consciousness on evolutionary phenomena, and some people see epigenetics as a way of doing it. It's annoying. Here's another example. There's going to be a conference in Austria this summer, organized by a philosopher who doesn't seem to know much about biology, to argue for a familiar creationist trope under a veneer of respectability. The idea that you can't get novelties or complexity from accidents, not even accidents post-natural selection. They have a host of mechanisms that they don't seem to understand that they've dragooned into supporting their claims, and epigenetics is one of them. Or, get ready for this, you should see how pop culture processes epigenetics. Would you believe you can reprogram your body with your belief system? Science says so, at least according to this person. Yeah, cow flops. This is just nothing but babbling bullpucky. So, however, I don't want you to get the impression that somehow these kinds of people discredit the whole field of epigenetics. They don't. There's a huge amount of research in epigenetics that is serious, that is science-based, that doesn't make these kinds of ridiculous claims like that you can sit down and meditate and change your DNA. That's not going to happen. But there are significant things that are happening in your DNA all the time. This is what I mean. Once upon a time, you were all just a single cell, a single zygote produced by fertilization via two gametes. And that single cell then differentiated in a whole bunch of different cell types like smooth muscle or bone or nerve cells or red blood cells. The process that produced that difference in gene expression in these different cell types is basically epigenetics. That what we have is a set of mechanisms that turn on and turn off genes in different ways in different tissues. This is necessary because all of these cell types in you have exactly the same genetic material, the same DNA, but somehow we have to regulate it so that some genes are turned on in smooth muscle, different set of genes are turned on in nerve cells. There are multiple ways we can carry out these kinds of gene regulation events. One mechanism is through transcription factors. So there are proteins that are produced that selectively bind to and switch on or switch off other genes. That's a transcription factor. So the body of transcription factors that you find in the cell are going to restrict and enhance the pattern of certain genes that are being active in that cell type. There are other mechanisms as well that are often more classically associated with what we consider epigenetics, mainly because they're more persistent. And that is, for instance, methylation. That one of the ways you can completely turn off a gene is by having a layer of methyl groups attached to the backbone of the DNA, which suppresses expression of that particular gene. There are other ways, too, that there are specific histones, these proteins that the DNA gets coiled around, that are more or less encouraging of gene expression. So you can have a class of histones with modified tails that do not allow for expression of a particular gene. In addition, there are also other fun ways to do this, like small interfering RNAs. There are RNAs that can be present in the cytoplasm that also act to suppress the expression of particular genes. Okay, this is where I'm going to get a little picky. So, as I've just shown you, there are cases of epigenetic transmission within the cells of a multicellular organism, and I consider that perfectly valid, perfectly interesting examples of epigenetics. Some people, though, seem to think that the only cases of epigenetics that count are those where you have transmission from one generation of individuals to another set of a generation of individuals, to the grandchildren and the great-grandchildren and so forth. That's fine. However, those mechanisms are exactly the same as the mechanisms we propose exist in cellular transmission of epigenetic information within a single multicellular organism. So it's kind of a distinction without any meaning, but that's where it begins to become interesting to evolutionary biologists, when you get multi-generational transmission of epigenetic information. So, for example, there's been some dramatic studies where we've seen the transmission of phenotypically expressed characteristics over multiple generations. So this is just one example. It's called transgenerational testicular dysgenesis. It's not a fun thing, but what's happened here is that an endocrine disruptor was given to a pregnant female mouse. And then in the next generation, she successfully had offspring. So in the next generation, what they observed is that the males had this phenomenon called testicular dysgenesis. That is, that they started out fine when they were young male mice. They were perfectly capable of reproducing, but as they got older, their testes basically disintegrated, just degenerated and stopped doing what they were supposed to do. Testosterone levels dropped and they were unable to reproduce any further. So that's testicular dysgenesis. Not surprising that treatment of a mother with an unpleasant chemical, a teratogen like an endocrine disruptor, has effects on her offspring. But what was surprising is that those males that successfully reproduced then had grandchildren that also expressed testicular dysgenesis in the males. And then when they reproduced, the great-grandchildren also expressed it. And then the great-great-grandchildren also had this same problem in the males. So that's a really interesting phenomenon. That's a persistent phenomenon. There have been other examples of long-lasting epigenetic effects. But let's consider a human example, and we do have some human examples. And here's the classic one. In 1944, the people in the Netherlands, that is about 4 million people, suffered great deprivation thanks to a blockade by Nazi Germany during the collapse of the Third Reich. It was a terrible event for the people of that country, but it was also a kind of experiment that can't otherwise be performed ethically. Thanks, Nazis. Although I should also add, the 20,000 people who died of starvation during this event probably weren't very thankful. Anyway, so we have data on people who suffered substantial caloric restriction, including pregnant women. And we also have good data on the month-by-month supply of food and the rations that were allocated to people. These people got as little as 400 calories per day, and thanks to meticulous record keeping, we also know about the fate of these people later. Unsurprisingly, women who were starved during mid to late pregnancy had children with low birth weights. Women who were starved in early pregnancy rebounded well and had children with normal birth weights. However, it was later observed that these individuals had significantly higher rates of obesity than their siblings. It has been suggested that their metabolism was reset by the early exposure during fetal development, and that predisposed them to inappropriate adaptation when plenty of food was made available later. What's most relevant here, though, is there's some evidence that the children of the children born during the hunger winter also had deleterious effects, suggesting that there was a transgenerational effect. Most interestingly, though, this cohort of people who experienced extreme hunger have been studied minutely to the point where we can look at patterns of methylation in single specific genes. So here's an example of that kind of analysis. These are some fairly complex graphs, so let me break it down step by step. So the green bars in each of these graphs represents the daily ration of food people received during the blockade. And you can see that it was rough for them. From December of 44 through May of 45, they were getting less than 1,000 calories a day, dropping as low as 500. Then in June of 45, the blockade was broken and food relief came flowing into the country. So you see this rapid rise. That's the recovery from starvation in June of 1945. There are two graphs here. The one on the left is for people whose mothers became pregnant late in the blockade. That is, they were embryos who were starved early in pregnancy. The graph on the right is for people whose mothers gave birth to them during the blockade. That is, they were embryos starved late in pregnancy. The data points are measurements of the degree of methylation of an important growth factor gene, insulin-like growth factor 2, or IGF-2, measurements that were made late in life relative to siblings who were not exposed to starvation. If starvation had no effect on this gene, then all the points ought to hover around the 0 line in gray, indicating no difference in methylation. And that's the case for the embryos starved late in pregnancy on the right. Their mean degree of methylation shows no difference from their siblings. On the left, though, we can see that if you were an embryo starved early, the methylation of your IGF-2 gene was depressed. Starvation led to hypomethylation. Now, these are really interesting data for a couple of reasons. One is that clearly fetal experience can have long-term effects on adult physiology. You grown-ups who are listening now have patterns of gene expression that are the subtle product of experiences you had in utero. This is something we have to hammer home, that you aren't just a product of your genes, but a product of genes shaped by environment. Another point, though, is these effects are individually rather weak. These mothers had to experience extreme deprivation at the hands of Nazis to see a slight hypomethylation of one gene. The individuals on the left had a reduction of about half a standard deviation from the norm. It seems to be real, but it's not overwhelming. Another point. Note the scatter in the data points. This is very noisy data. Part of the reason for the wide variation has to be because there are a great many factors that influence these methylation events. If being starved early in gestation can depress IGF-2 methylation, Who's to say that other unquantified inputs can increase IGF-2 methylation? What the environment can take away, the environment can also give back. So one problem with discussing epigenetics and evolution is that it is a transient and malleable phenomenon. That an environmental influence may have a multi-generational effect adds a level of inertia to the evolutionary response, a kind of hysteresis in the system. But it's still going to require a genetic change to make it more enduring and relevant to evolutionary theory. So there's another problem with epigenetic transmission across generations. And this is an old story. What you see here is a tree diagram by ViceMoms from 1893, illustrating the pattern of divisions and fate decisions in the nematode Rhabditis. So I know it's poorly marked, but endoderm is at the bottom left, ectoderm at the bottom right, mesoderm is top center with the germline flanking either side of that. What you see here is the tree-like pedigree of the cell fate decisions that make up the tissues of the animal. What happens is that the parent cell, the node of each cell type, activates specific switches that commit it and all of its descendant cells to a particular class of tissue. That is, there is a cellular transgenerational commitment of the cell lineages within the animal that gradually narrows the range of fates that cells at the terminal lineages can follow. Is epigenetics important in development? You betcha. It shapes developmental decisions and makes you a well-organized assortment of different kinds of cells. But what we're concerned with now is, does epigenetics allow for transgenerational transmission of patterns of gene expression between generations of individuals? And Weissman had an answer for that, too. What Weissman noted is that there's a segregation of cell types into two major classes of tissues. The somatic cells, which make up, for instance, your pancreas and liver and brain and most of your body, and the germ cells, which make up a nearly invisible small population of reproductive cells in your gonads, your testes, and your ovaries. And he postulated what was called Weissmann's barrier, that what happens in your pancreas stays in your pancreas, and no pancreatic cells get to cross the barrier, enter your gonads, and be transmitted to your progeny. This is a death knell for Lamarckian inheritance. I'm sorry, guys, no matter how hard you work out and how swole you get, you're not going to inseminate your spouse with your muscle cells, but only your sperm cells. This means that Lamarck is not a viable model for epigenetics either. So I have to explain why that is so. So let's take a look at a really simple model organism. So what I've got here is a cartoon of a 7-celled animal, and it's got various cell types like liver and gut and bone and brain and muscle, and of course gonad, because it's going to need to reproduce. Each of these cell types has its own set of epigenetic marks. They've all got the same DNA, they've all got the same genetic information, but they are expressing it in different ways because their patterns of expression have been modified by these epigenetic factors like methylation and transcription factors and so forth. So that's our starting point. We've got this organism. That organism had to have arisen from a single-celled zygote. So that's our first step. And that single-celled zygote would have had a pattern of gene expression that was different from all the cells that are now expressed in the adult organism. And that zygote would have gone through embryogenesis. It would have developed in such a way that it would have had complex shifting patterns of gene expression as well in all the cells of its tissues as it was forming those tissues. And then eventually it becomes an adult, like we showed in the previous diagram. Now, if this is your typical diploid, sexually reproducing multicellular organism, another organism might come along. So let's say that's a male that comes along. And then both individuals are going to produce gametes. Notice I cleverly colored them pink and blue to make it clear. So these are gametes that also have their own unique patterns of gene expression, their own methylation states, their own complex organization of regulatory factors in their genomes. Also, I'll mention here that male and female gametes also tend to have different patterns of epigenetic marks, which is very important in the phenomenon called imprinting, which I will not be discussing here. Then those two gametes come together and they fuse to form a zygote. Notice the gametes had their own unique pattern of gene expression. The zygote's got its own unique pattern of gene expression. They're different from the adult. And we go once again through a pattern of embryogenesis, which will yet again have a different pattern of gene expression in all of these tissues to produce another adult. The cycle begins all over again. So I think you might see a little problem here. If you want to carry out the Lamarckian transmission of, for instance, changes in your muscular tissue or your brain tissue, the problem is that when you produce a gamete, you're going through an epigenetic bottleneck, that you are now producing an organism that has to reset all of its epigenetic marks to a gametic or zygotic state. So it's going to turn off bone-specific marks, for instance. It's going to express new epigenetic marks. And then it's going to go through a complex pattern of development. It's going to reset them yet again. There is no way for very many of these marks to survive this transition from the adult to the gamete to the zygote. So this is a scheme that isn't going to work very well. In addition, notice that there are, I illustrated a fake adult with 7 different cell types. You have many, many more cell types than that. You have hundreds of cell types. There is no way to take all the epigenetic information in all of those different cell types and transmit it through a single-celled zygote to the next generation. So there's got to be a massive reduction in the information present in that zygote. Okay, gang, let's put this all together. So the case I'm making is not that epigenetics is unimportant. The case I'm making is that it's really difficult to make a case for the role of epigenetics and having a significant effect on evolutionary pathways. And there's a couple of reasons for that I'll mention here. One is that what we've seen is that the effects are variable and weak. Now, that is not a dead-on case against the role of epigenetics, because I would also say that many genetic effects are also variable and weak. However, it means that it's really hard to analyze and hard to predict. And it's also the case that we have a multitude of different kinds of epigenetic effects that might be modulating these responses. A bigger problem in my mind is that it's unlikely that most phenotypes produced by epigenetic marks could overcome epigenetic bottlenecks. That there's no way for us to transmit in a Lamarckian fashion, as some people are prone to suggest, information from the various tissues of your body into the germline. So that's just not going to happen. Another important thing is that what seems to be going on here, at least to me, is that the key role of epigenetics is in plasticity. That what it is a way for genomes to adapt to new environments fairly rapidly. That means, though, that response has to also be present already in the genome, and we're just selecting for patterns of expression, which in itself is kind of an interesting idea. Theodosius Dobzhansky, for instance, years ago suggested that maybe what's going on here is that we are looking at selection of reaction norms, not simply selections of phenotype, which I find a kind of provocative suggestion, although in some ways kind of hard to analyze. Another concern I have is statistical bias. That what we're often doing in these epigenetic studies is looking at a random input and then asking what comes out at the other end. For instance, in the hunger study, another thing that they've observed is that there's an increased frequency of schizophrenia in the next generation. This was not a prior hypothesis. There's no reason to predict that starving a fetus could increase the frequency of schizophrenia in your grandchildren, for instance. So that means that it's kind of problematic statistically to do these analyses. What would be better is to have a direct link to the genes responsible for a phenomenon and ask if they are differentially modified by the environmental stimulus you're looking at. I think the studies of IGF-2 are a good example of how to do that right. So to conclude, I would say, the mechanisms of the neo-Darwinian synthesis are still going strong. Mutation and recombination are the primary sources of evolutionary and phenotypic novelty. Other phenomena, for instance, Evo-Devo, I'm keen on Evo-Devo, things like epigenetics, are real and important, but they don't supplant known, tested mechanisms of evolution. They enhance, but they do not substitute. There is zero evidence for any kind of Lamarckian effect of epigenetics. And finally, I would say beware the hype. There is no epigenetics revolution. There is a whole bunch of really cool research, good science being done, but let's not pretend that it's going to replace evolutionary theory at any time in the near future or the distant future. Okay, thanks, and we'll talk to you next week.